Hi class, and welcome to Module 6. Now we already told you about plant propagation, sexual plant propagation using seeds. So what we're going to cover in this module is vegetative propagation or asexual plant propagation. Now, when you're doing plant propagation vegetatively, one of the things that you really want to have is a good resource to look at, because you don't want to go through and try something that someone else has already done and doesn't work. So these are three good resources that I use that tell you with particular plants what is the best way to propagate them. Now some of these will tell you that the best way and the only way is using seed, and then others will tell you doing cuttings, the different types of cuttings, what works best for particular species of plant. So what I like to do before I propagate anything is look it up in one of these resources and see what someone else has already done, because if they've done it and been successful, I don't want to experiment with it, because what I really after is results. Now during the course of this presentation, we're going to mention rooting hormones several times, using those. And what are rooting hormones? Well, you can see that the rooting hormones, they're simply auxins. That's natural plant hormone, and it's involved in cell elongation and root initiation. So this hormone will actually help the cuttings that you take develop roots faster. And you really want to use it if you have slow or difficult to root cuttings, or if it's one that you aren't sure if it needs it or not, go ahead and use it, because in most cases it can't hurt. And what this will do for you, it increases the uniformity and speed of rooting by using these root hormones. Now there are two different types of rooting hormones. There are powders and then there are liquid forms. The powdered form, you want to dip the basal end of the cutting into the powder. You don't want to dip your cutting directly into your container of rooting hormone, because that's going to cause possible contamination problems. Anything that's on the cutting will end up in your rooting hormone powder. So what I like to do is break out little bits of it into other containers. You can pour it into the little cap, or just pour it out on the piece of wax paper. And you can still dip your cutting into the rooting hormone, and that way you don't contaminate your whole container of hormone. And you don't want to get the powder on there super thick. So what I like to do is, once I've dipped it, kind of tap the cutting on the edge of the table or something like that to knock off any super excess hormone powder, and then make a hole in my media and stick the cutting into the media and then pack up around it. Now you can also get rooting hormone in the liquid form. This is the hormone, and it's usually at 500 to 1,000 parts per million. Here's a couple of hormones that are available, the Dip and Grow and the Rhizopon AA. Those are brands of commonly available liquid rooting hormone. And what you want to make sure with the liquid and with the powder is you don't get any of it onto the leaves of your cutting. You want it only to be on the area where you want roots to develop. And with the liquid, you want to make a solution of it into a smaller container. Now usually, I know with Dip and Grow and some of these others, they come with a little container that tells you how much to dilute out the concentrated solution that you have. You can use that small container to do your dilution in, dip all your cuttings, stick them into the rooting media, and then you can dump out whatever is left over and wash out your small container, and you don't contaminate your concentrated bottle of liquid rooting hormone. Now there are several advantages to doing vegetative propagation. One of them is you can produce plants faster. You produce a lot more plants a lot faster. If you think about the fact that you can take a small bush and take hundreds of cuttings off of it and root all of them, you can have one, quote, mother plant, and you will produce hundreds or more of cuttings from that mother plant. And by doing vegetative propagation, you're going to make plants that are virtually identical to that parental type, to that mother plant. So you don't have to worry about genetic changes or hybridization or any of that, because you're essentially cloning that mother plant. So that is a real good advantage to using vegetative propagation. And a third advantage to vegetative propagation is that you get the plants, when they're rooted and ready to use, they're already in the mature stage because you're starting with mature tissue from the mother plant. So they don't have to go through that juvenile stage as they would if you started with seeds. This really shows it's in some of the fruiting trees and shrubs that we have, because if you start from seed, it may take 10, 15, 20 years before your tree starts to produce fruit. But because they do another type of vegetative propagation, grafting, they're using old tissue on young roots. And that old tissue is mature, and as soon as it starts to grow, within a couple of years, you're going to start to get flowering and fruit from that fruit tree, whereas it would take 15 years if you start from seed. There are several methods of vegetative propagation, and here we're listing the main ones that we're going to talk about. We have division, layering, using storage organs, grafting, tissue culture, and cuttings. Now we're going to talk a little bit about all of these, but we're going to concentrate mainly on using cuttings, because that is the simplest, the easiest, the most often used method by gardeners, is using cuttings. Now with divisions, that's just the separation of one plant into several self-supporting plants. Usually you can do those divisions using a shovel or a knife, just by going into what we have here is rhubarb. You can take the shovel and stick it in between those crowns that are coming up, push it into the ground, cut those out separate from the mother plant, and then you can transfer those to wherever you want, into a container, into another location in your garden, and you have new plants. Now, the divisions that usually occurs with herbaceous perennials is where you most often see that, and it's most often used. And like I said, one of the edibles that divisions is used to propagate is with rhubarb. Now we want to talk about layering. Now layering is simply where plants form new roots, and even entire plants, where the stems of the mother plant come into contact with the soil or the medium that you're growing them in. There are multiple types of layering. They all have similar results and similar approaches to developing that layering process. But for technical purposes, they're usually broken down into air layering, simple layering, tip layering, trench layering, and mound layering. Now with air layering, what you want to do is select a stem that's about pencil size. Then you come back from the tip, maybe 12, 15 inches. You clear away all the leaves and branches around about a 4, 5, maybe 8-inch section of the stem. And then with a sharp tool, you want to make cuts parallel to each other, circling the stem, as you can see in this slide. Now you want to make sure you're cutting only through the bark area of the plant and not completely through the xylem, because the plant is still going to need, that tip of the stem is still going to need water. It's still going to be producing food for itself, but it needs water and the nutrients coming up from the soil. So air layering, that's one that if you do it, it'll probably take a little bit of practice to get it perfectly correct, but just want to move that outer barky area and expose the cambium. And then once you've done that, you want to remove that layer, and then what I like to do is dust that area with the powdered rooting hormone. Now a lot of these you can do without dusting that area, and they will root just fine without the rooting hormone. But like I said, a lot of times it does help. And so dust that area that you've opened and removed the bark from with a little bit of the rooting hormone. And then wrap that with moist sphagnum moss. Now you don't want it soppy wet. You just want to soak it really good, get rid of the excess, and then wrap that around that area where you've removed the bark and put the rooting hormone. And then wrap that sphagnum moss in plastic, because you want it to be watertight. You want it to stay high moisture in there. And then with twist ties, I like to use ones with the wire going through them and plastic on them. You want to twist tie around below the cut mark and above the cut mark on either side of that little bundle that you've created. So you want to put on the twist tie, and then you just leave it. And after several weeks to a month, depending on the plant that you're doing, roots will develop inside that little package, and you're ready then to take your new plant from it. So using very sharp pruning shears, once you see you've gotten roots developed, you remove the plastic. Don't really need to shake off the sphagnum moss, and then cut just below where you made those incisions to remove the bark. Right below where the roots are developing, cut it off, and now pot up your new plant. You have a new plant that you've created by the air layering process. And there are several edibles that can use air layering to make new plants. Citrus, apples, pears, peaches. A lot of the fruit trees have been successfully air layered. And so this is another way of making vegetative propagation of your fruit trees using air layering. The second layering example is just simple layering. Now with this, you choose a low hanging branch, again about pixel width, and remove all the leaves in the area that you're going to bury underground. What I like to do is to wound that underside of that branch that's going to be buried in the ground just a little bit. You make small thin slits using a sharp knife, a sterile sharp knife, and then you want to dust those slits with the rooting hormone, and then bury that underground. You can even use a staple or some sort to hold it into the ground whenever you bury it. Then cover it back up, and then I like to use a little stake to bring the tip of the limb back up, pointing straight up. That makes it grow straight and gives me something to watch as the roots are developing. And once the roots have developed, you just cut this loose from the mother plant, dig it up, and then transfer it to a new location or into a container. And this simple layering works great. It works great with blueberries, is one edible that it just really works well with. And then there's tip layering and trench layering. Now these, like I said, layering again is simply getting the plant to develop roots on a stem that while it's still attached to the plant. These two, with brambles especially, blackberries and raspberries, the tip layering works really nicely. They even do it naturally. And another plant that does it naturally that's an edible is strawberries. That is tip layering that's happened whenever those strawberries are making those little baby plants next to the mother plant. And with tip layering, you simply bury the tip of a branch underground, and then the apical bud will grow upwards to produce the new plant, and because it's underground in a moist environment, it will develop roots. And like I said, there are some plants that this works really well on. There's other plants that you can bury the tip of the limb and it's never going to make roots. So once again, go back to those resources that you have and see what works best for propagating the different plants you're going to do. And with trench layering, it's simply doing multiple branch layering because you're burying an entire branch underground and letting each bud, axillary bud, on those branches make new plants. So you can get lots of little plants coming up and then cut all those loose from the mother plant and transfer them into your container. And the final layering method is mound layering. And this one's used with shrubs primarily. It can work with blueberries, again, like with the branch layering. Blueberries can do mound layering. And with mound layering, what you want to do is to cut back the mother plant to within an inch or two from the ground. And when you do that, it's going to start to form new branches coming up. So you cut it back during the dormant season. Then whenever spring rolls around, it's going to start producing all these new shoots. And you bury that part of the stem underground in a mound, as you see here in the illustration. So you mound that soil over the new shoots as they begin to grow, and then roots will develop along the stems of those new shoots. They'll begin to form adventitious roots because they're underground, they're in a moist environment, and they will start to form roots. So once you give it a couple of months for all this to take place, you remove the soil that you've mounded up, and you can easily cut away using, again, very sharp pruning shears that you sterilize. Cut away each of those new individual little plants and transfer those to a potting medium in a container, in a raised bed, or another location in your garden. And by doing this, you can make 10, 15, sometimes 20 plants from a single mother plant just by using the mound layering process. Okay, another method of vegetative reproduction is using the storage organs that the plants naturally make and using those to produce new plants. There are several storage organs. The first one we're going to talk about are bulbs. Now, there is a true botanical definition for what a bulb is. In your catalogs, most of the plants that you order, some underground portion of it to plant and to produce new plants, your irises, your daffodils, all of those, the lilies, they'll be generally termed bulbs. However, a true bulb has a short stem and fleshy leaves at the base. And it's these fleshy leaves that have been modified as to storage organs that create the bulb. As you can see here, and as we learned in botany, this little basal plate or basal stem in an onion, that is the stem. Then all these, which we call scales, are modified leaves, modified to be storage organs. The roots will develop down here, and actually this center, which if you keep onions too long, you'll see it start to develop. This is the apical bud right here. And this will grow up into the onion stalk if you leave it long enough or if you plant it out. So what we got here now is a true bulb. With these, you can just plant these into the ground and they will produce a new plant. And as you can tell from the example I have here, the primary edibles that we reproduce using true bulbs are in the allium family, onions, leeks, shallots, and garlic. These are true bulbs, and we reproduce plants from these vegetatively using their storage organ. Another storage organ are corms. Now, corm, that's just a short, vertical, swollen underground stem that serves as a storage organ. Right here in this example, you can see we have the stem tissue. The apical bud is right at the very top. So this is all swollen stem tissue right here, which stores energy. The roots develop down here, and then the outer layer, the sheath of the corm, it's termed as a tunic. And you can plant these in the ground. They'll produce new plants from it, and they will also then, on the side, produce new little corms, sometimes termed cormels, and these are just small corms that will develop into plants as well. So once you've taken the large one here that we have in this picture, once it grows up and produces a plant, it's going to make little side cormels here that you can collect and propagate your plants that way. One of the ones that is propagated this way is celeriac. As you can see, the large corm here, that's the edible part of celeriac. Taro is another one, and water chestnuts. The ones that you usually see have had all of the tunic removed and the roots are gone, so all you're really getting is this little stem part, which is actually comes from the corm. That's what water chestnuts are. Another storage organ are the rhizomes. And rhizomes are horizontal underground plant stems, and they can produce new shoots and root systems at different buds on the on the rhizome. So this is a rhizome. You can see a new plant growing up. It's an underground stem, so you do have internodes. And these little marks on it here are the nodes. And at the tip of this one, you can see there is an apical bud. And from that, a new plant will grow up. Roots are developing down here. With rhizomes, you can cut them apart into pieces and plant each individual piece. It will produce a new plant. And commonly reproduced plants that are edible that we have that are reproduced by rhizomes are ginger and turmeric. Both of those produce rhizomes and are reproduced by using rhizomes. Now there's another underground stem, but it's not really an underground stem. It's the above ground stem, which are stolons. The primary difference between stolons and rhizomes, they're both horizontal stems. Rhizomes grow underground. Stolons creep along on top of the ground. So if you ever see the term stolon, that's what it's talking about. It's above ground horizontal stem, whereas the rhizomes are underground horizontal stems. Other storage organs, there's two types of tubers. There's root tubers and stem tubers. The root tubers, that's just a swollen section of the root that is modified to be a storage organ. And you can see on our diagram here, the stem is underground, and it gets this, now this is a root coming out, the root stalk, and it's formed this swollen area here, which is the underground root tuber. And the most common one that we know that's edible is the sweet potato, or yams, which is slightly different from a sweet potato. Both of those are underground storage organs or root tubers. And from these you can produce new plants. You have the distal end and the proximal end, which is the end attached to the root stalk. And inside, if you look at it, it looks just like a root morphologically. And this is a nice way that I'm sure a lot of you have done at home just for the fun of it. Sweet potatoes will actually start to form stems if you store them too long. And you can just take a sweet potato. Now with this, you want to make sure, why I pointed out there's a distal end and a basal end. If you're going to do this type of experiment, the part that goes into the water is the basal end. And the part that's going to form slips or new shoots is the distal end. So you can put a sweet potato into a jar of water like this. This one has toothpicks holding it up, so the whole thing isn't underwater. Roots will form down here, and stems and new shoots will form here. The second type of tuber is a stem tuber. This one you've already been told about in the botany class. A stem tuber, that's just a swollen underground stem that has developed into a storage organ. The most common one we know that's edible is the Irish potato or the white potato. As you can see on this potato laying here, this would be the end that's not attached to the stem, or this would be the basal end, this would be the distal end. We have an apical bud, and then all these eyes on the potato, those are actually axillary buds on this potato stem. The little tiny dots on it, those are lenticels, and every eye, if you look at it closely, it has this little smiley face on it, or half moon, or crescent moon look below the eye. That's actually a scale leaf. That is a modified little leaf on the potato tuber. And so with white potatoes, you can take these and cut it into pieces and make sure there's an eye on every piece that you cut. And then there's two ways that you can reproduce that. You can store them for a small length of time until the cut area calluses over, and then just plant it. It will make new potatoes, the stem will come up, the roots will develop and go down, and you'll have a new potato. Some people go ahead and plant them directly as soon as they cut the potato into the ground. The only hazard with that is, since it's a fresh cut, the likelihood or the probability of getting fungus infecting the tuber piece that you put in the ground and rotting it before the plant has a chance to develop is higher. If you let it callus over first, your success rate will be better. Now, grafting. That's another way of doing vegetative propagation. And with grafting, all you're doing is joining two pieces of separate plants together to form one plant that's going to function as a single plant. Most grafting, you have the root portion of the plant from one plant, and then the top portion, termed the scion, from the other plant. So you have a scion, which is the upper portion, and the rootstock, which is the lower portion. Now with grafting, there's a lot of different methods for grafting, and grafting is kind of a skill, a science, and an art. I work with people doing grafting and doing identical methods, identical approaches, identical procedures. One person can get 95 to 100% success rate grafting, and the other person's getting maybe 20, 25%. There's no apparent difference in what they're doing, but there's something a little bit different. It's kind of like following your mom's recipe, and she didn't write down that she uses a cup of something plus just a little bit more to make the consistencies right, and all you're doing is using a cup and you never get it right. So it is a science in addition to being an art and a skill. Now with grafting, it's a little bit more labor intensive, but the advantages of grafting is you get the disease resistance of the root system and the horticultural benefits of the scion system. So the scion is giving you the above ground qualities that you want. The root stock is giving you the below ground qualities that you want. So a lot of times you'll increase your disease resistance, and you can even control the size of the scion based on the root stock. A lot of dwarf fruit trees are dwarfed because of the rootstock they use. The scion or top portion, if it were on any other rootstock, it would grow into a full-size tree. But because it's on a rootstock that has the qualities of dwarfing the scion or above ground portion, you actually develop a dwarf fruit tree. A tissue culture is another method of plant propagation that's vegetative. A tissue culture, that is simply culturing the plant's seeds, its organs. Explants are just little pieces of plant taken from another plant. Plant tissues, cells, or even protoplasts, and you grow them on a chemically defined synthetic media. As you can see here, these are growing in test tubes on a synthetic medium, which is providing the plant the different nutrients and the different hormones it needs to develop into a new plant. The stem and leaves and everything developing here. The roots are developing here. And this is all done under a highly controlled environment, controlled with the light, the moisture, the temperature. Tissue culturing is much more technical, much more involved, requires a lot more expensive equipment. We're not going to spend any more time on tissue culture or grafting. Hopefully in the future we can offer another course on just grafting for you. So if anyone's really interested, watch out for that course coming in the future. Now we're going to move to cuttings. Now this is the most often used procedure by home gardeners for increasing plants vegetatively. And most plants do have the ability to regenerate entire plants just from a small piece of tissue or even from one single cell. The term for this is totipotency. The plants are called totipotent because they have the totality within that single cell or that single piece of tissue to produce an entirely new plant. And we take advantage of that characteristic of plants to produce new plants vegetatively. And as far as we know, so that this ability to reproduce an entire new organism from just a piece of tissue is limited to plants. They are the only ones that you can do this with. Now, with cuttings, the idea is that once you have a mature cell, plant cell, any type of mature cell, It's really difficult to get that mature cell to initiate new growth into an entire plant. So with cuttings, what we're actually doing is taking advantage of the meristematic tissue, or the tissue within a plant that is as yet undifferentiated, and the cells are dividing. And the meristematic tissue in a plant can divide indefinitely, producing new cells. And the meristematic tissue in plant cells is found in the shoot tips, in the root tips, and in the vascular cambium. And it's this meristematic tissue that we're taking advantage of to produce entirely new plants. Here's some micrographs of the different meristematic areas in a plant. Here's the plant itself, and then here's the apical meristem. And this area right in here, you have the developing leaves, and this meristematic tissue is constantly dividing, growing forth, and the cells are differentiating into the different parts of the plant, into the stem cells, the xylem cells. All that's happening once the cells divide. This is the apical meristem of the root, and this will always be found at the root tips. This is constantly growing, dividing, and producing new roots. But this meristematic area in the shoot and in the root is where cell division and differentiation is taking place. Another meristematic area is the vascular cambium, which in dicots surrounds the entire stem of the plant. In monocots, it surrounds the individual bundles of vascular tissue within the plant. So in the air layering that we showed you earlier, and in all the layering procedures that we showed you earlier, with those, what we're doing is taking advantage of this vascular cambium that exists within the plant. This area of undifferentiated cells is where the new roots, the new shoots, and everything will develop from this cambium, which is the vascular cambium. Now, there are several types of cuttings that you can do. You can do stem cuttings, you can do leaf cuttings, and you can do root cuttings. Remember from botany, here's our stylized plant. Below ground, we have the root system, the primary roots, the lateral roots, the root cap, the root tip, and actually on our lateral roots, those each actually have a root tip that has meristematic tissue in it as well. Then we have the stem, and we have the different buds, the apical bud, the axillary buds or lateral buds. These all have meristematic tissue in them. And then we have the leaves, so we can see we can take stem cuttings, we can take leaf cuttings, or we can take root cuttings. And there are different types of stem cuttings. You can have softwood cuttings, and this is usually with hardwoods, it's the spring succulent growth, or with herbaceous plants, they are all essentially softwood cuttings because they never form hard woody stems. Then there's the semi-hardwood cuttings, and those, you would take those cuttings in the summertime once the stem begins to harden. Softwood, it's very soft and fleshy. Semi-hardwood, it's begun to lignify and become hard. And then there's the hardwood, which comes later on. And this is usually the stems are at least a year old. So the plant has had time to mature and fully lignify that area of its stem. It's very hard, not very bendable. And this would be where you would take hardwood cuttings from. And here on our little stylized plant, you can see this area, which is the new growth, would be where the softwood cuttings would come from. And then down here, as it's begun to harden, this would be where the semi-hardwood cuttings would come from. And then this year-old or older area of the stem, which is really hardened and lignified, this is where the hardwood cuttings would come from. And the softwood cuttings, that's what mostly we deal with vegetable plants and with edibles, is doing the softwood cuttings. The best way to take softwood cuttings from vegetables, or if you're going to be propagating some of your other plants, there are a lot of plants that you can take cuttings from like this. Go back to your resources. There might be some really nice ornamentals in your garden or in your neighbor's garden that you'd like to have in your garden, and you can do cuttings from those, and you know that what you get when you do this, it's vegetative propagation, and you're going to get the exact same plant that you started with. So with softwood cuttings, you take those early in the morning. You want to put them in a plastic bag or some type of sealed container so that they don't dry out. And what I like to do is immediately put them into some cooler. I'll put ice in it to keep it cool. Even if you just have a cooler with no ice in it, it's going to be a lot cooler than the outside temperature, because you don't want these drying out. So put them into the cooler and then take them back to your rooting area. You'll always take your cuttings, no matter what type you're taking, with a very sharp knife or very sharp pruning shears. You want it to be sharp so that you don't crush the stems as you're taking the cuttings. Because if you crush the tissue, a lot of times you won't get the development of the roots on the stem because you've destroyed the openings of the vascular tissue. So the top portion of the stem that you save can't get the water up into it that it needs. And so you don't want to crush the stem. You want to slice the stem. So make sure you're using very sharp knife, very sharp pruning shears, and if you can, sterilize them. You can do this by having some rubbing alcohol with you to dip it in before you make the cuts. Dip it in it, let it evaporate, then make your cut. Or you can dip it into a 10% Clorox solution before you make the cut. Dip it into that solution, let it dry, and then go ahead and make your cut. And by doing that, you don't introduce disease into your cutting or into the mother plant that you're taking the cutting from. So you take your stem cutting, the softwood cutting, dip it into the rooting hormone like we told you before, either the powder or the liquid, and then make a hole in your rooting media and insert the cutting into the hole. This is really important to make the hole first if you're doing softwood cuttings or herbaceous cuttings, because if you try to push the stem into the rooting media without making the hole first, a lot of times you will crush the stem or break it off, and now you've lost your cutting. With hardwood cuttings or semi-hardwood cuttings, what you want to do is take a cutting, like I said, that's begun to lignify, and the axillary buds have already formed on this part of the stem. That's another way to tell that you're using a semi-hardwood cutting. And with me, I like to cut just below a node. Once again, using a sharp, sterilized cutting utensil. Trim away the leaves except for the ones on the very upper end of the cutting. And if you have large leaves, you'll want to trim those as well. With gardenia cuttings, when I take those, if it has a really large leaf, I'll cut the leaf in half or even cut more of it off and leave just a small portion of the leaf left attached to the stem. You want to reduce as much above ground leaf tissue as possible, because that's where transpiration and loss of water will take place. And so you don't want that happening with your cutting. But also, you want to leave a little bit of the tissue so photosynthesis can still continue. So you leave a little bit of leaf tissue so the plant can still make its own food while it's developing roots. But you don't want to leave so much that the plant dries out during the transpiration process because it has such a large leaf surface. So go ahead and trim away some of these leaves to reduce the moisture. And then looking at your resource, it'll tell you whether wounding the bottom or basal portion of the cutting enhances root development. With some plants it does, with other plants it doesn't. And so with my gardenias, sometimes I'll make little slices in the end, and that enhances the development of the roots on the stem end. Dip it into rooting hormone, and again, if you just get into the practice of doing this, you never have to think about it. Make a hole, and then insert the cutting, and press the rooting medium back around the inserted cutting. And with hardwood cuttings, these are ones that we never really deal with when we're talking about vegetables. These are always herbaceous, but if you're going to do some other plants, ornamentals, or even some of the fruiting plants, fruiting trees that you can do hardwood cuttings from. And with these, you want to take 1 year, 2 year old stems You don't want it to be too old so that the bark has really gotten tough on it, and you definitely don't want it to be too young. And if you wait too long, the stem starts to get really thick and wide. So usually we're dealing with about pencil size diameter or less when we're talking about taking hardwood cuttings. And you take the cutting by removing the semi-hardwood material from the portion of the stem that you select to make a cutting from. You discard that and then cut the hardwood cutting that you're going to use about 2 to 3 nodes below where you just cut off that semi-hardwood portion. Wounding with hardwood cuttings is usually more required than with semi-hardwood cuttings. So go ahead and wound the end, dip it in your rooting hormone, make a hole in your media, and stick it into the rooting media. Now, usually once you've done these, depending on the plant and the resources will tell you, it can take anywhere from 3 weeks up to 6 months or longer, depending on the plant, for the roots to develop. So check that out in your resource before you do it, so you know about how long it is you want to wait before really checking out your cuttings and see if they develop roots. Another way of making vegetative cuttings is using leaf cuttings. And the leaf cuttings can be just a blade by themselves. It can be a blade plus the petiole. It can be sections of a leaf blade. And you can also do what is called a leaf bud cutting. And with that, you're taking the leaf, its petiole, and a small portion of the stem that contains an axillary bud. Now, these are the different types of leaf cuttings you can make. Here are diagrams showing the different ones. This is the whole leaf blade itself. And this is a very often used method if you're taking the leaf blade, is by taking the leaf and then you cut the veins on the leaf, makes little slits in the leaf across the vein at different portions. By doing that, you're taking advantage of some of the meristematic tissue in that plant leaf, and then lay it flat on your rooting media. And when you do that, little plants will form at each of those incisions you made in the leaf. So this is using an entire leaf, using a leaf plus the petiole. With this, you're burying the petiole portion of this cutting that you've taken below ground, below your medium, and the roots will form on that petiole. And then it will develop a new shoot coming up from that petiole as well. So the leaf, what it's doing is providing photosynthetic products to the newly developing roots and stems. And so this is the photosynthesizing portion of the plant as it develops. Roots will form down here, and a new little plant will grow up from here. Using leaf blade sections, which is what we have here. Now this is very similar to using a whole blade and making little slices in it. What you're simply doing with that is you're taking the leaf and cutting it into sections or portions. Now this usually works well by taking the section and then standing it horizontally in your rooting media to allow the roots to develop down here, and the new little plant will grow up this way. So this uses a whole leaf, but then you're cutting it up into sections. It works great with leaves that are palmately venated, have palmate venation, because you can cut following a vein back to the main part of the plant. You're actually slicing the petiole and then sticking that into the rooting media and developing new plants. And then this is the leaf bud cutting that we're showing here, where it has an entire leaf, and then you remove this portion from the stem, which still has the little bit of bud here. It has the axillary bud, and you stick that into the growing medium. Now the axillary bud is actually what is rooting. Roots develop, of course, below ground. The axillary bud will grow upward, produce the new plant, and the initial photosynthesis is being done by the leaf blade that you still have attached to this axillary bud. And there's a phenomenon known as blind cuttings, which happens, one common thing that I've seen it with a lot is with the rubber tree, where you can take the entire leaf with the petiole, and then you do a cutting like this, like the leaf and petiole cutting, put it into your rooting medium, and the petiole will actually form roots. But that's all it forms. So you have a leaf with roots, and it never develops a new plant. And with this can actually live for several years. Just a leaf sticking in the ground, it's got the roots, so the roots are bringing all the nutrients that the little leaf needs up to it. So photosynthesis is taking place. The nutrients and water are moving up into the leaf. The leaf is making the sugars and food, sending them back down to the roots. And so essentially you have a rooted leaf that never develops into a plant. That's why it's really important to check your resources, because with a rubber tree plant, you can get this to happen and produce roots, but you're never going to get a plant back from it. If a plant really ever develops, it's probably because when it was ripped off from the plant, there was a little bit of meristematic tissue still attached. So these are called blind cuttings. They develop roots, but they never develop into a plant. And then there's root cuttings. And with root cuttings, what you're actually doing is taking pieces from the roots of the plant and using those to produce new plants. Now, those root cuttings, we usually take those, or like to take those, in late winter or early spring while the plant is dormant, or as close to being dormant as possible. And one of the main reasons for taking them and when the roots are dormant and the plant is dormant is because during the dormancy phase of the plant's life, it's storing a lot of starches and other food in the root system. And when it's actively growing, there isn't very much stored in the roots. So by taking the cuttings during the dormancy period, you're increasing the chance that the plant that develops from your root cutting is going to survive. So you dig up the parent plant, you have to do that, and then you cut a 2 to 3 inch piece of the root tip. Or you can even take a section of the root. It doesn't have to be the root tip itself. Once again, using sharp knife pruners and that are sterilized, very sharp. And as soon as you take those root cuttings, go ahead and repot or replant the parent plant. You don't want it staying out of the ground because the roots are going to dry out. So take your root cuttings, put them in a plastic bag to store them. Labeled plastic bag, of course. Label, 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 everything. When you're taking cuttings, label your bags. Do everything with a label. And then you go ahead and replant your parent plant. And then there's a couple of different methods you can do to get these roots to produce plants. You can lay them horizontally on a moist rooting medium and then cover it with more media, or sometimes if you have a high humid area where you're doing your rootings, you don't even have to cover it. I like to cover mine always because it gives a better chance that they're not going to dry out. And remember now that the shoots of the plant are going to grow from the cut end, or if you've taken a root section, the end closest to the main root that you cut it away from. That's really important because if you have really thick pieces of root, you can also stick them in the rooting media vertically. But if you put them in upside down, chances are you're not going to get a plant from it. So you want to make sure that you have the root tip end of the cut section underground and the stem end portion of the root above ground. And by doing that, your plant will develop roots and new stems. Really important to make sure you know which end is which when you're doing these root cuttings. Now, we've been talking a lot about rooting media and putting your plants into rooting media. Now, rooting media is different from standard potting mix. There are important things about rooting media characteristics that it has to have if you're going to be successful doing these cuttings. The media has to be able to hold moisture, but not be waterlogged. So therefore, it needs to provide good aeration. Remember, roots need air as much as they need water. So you cannot do it in a soggy solution. So your rooting media has to provide aeration, has to hold moisture, and whatever you got your rooting media in, it has to provide good drainage, because you don't want them to be soaked while they're rooting. And the fourth thing is you want it to be disease-free and weed-free. So starting with new sterile material, which most potting mixes are, or whatever we're going to make our rooting media out of, comes to us fairly sterile. So you don't want this weed seed in there, you don't want disease in there. So you want it to be as disease-free and weed-free as possible. And some of the commonly used rooting media is peat moss and perlite. What they do is they provide good moisture retention and good aeration. And then you can use vermiculite. That gives you good moisture retention, but it's a lot finer than perlite, and so the aeration isn't quite as good. With sand, you can add that to rooting media if you need to increase the aeration, because sand will provide really good aeration in your rooting media, but it has no moisture retention capacity, so it's going to dry out on you really fast. A good mix that I like to use also is a mixture of sand and peat moss. So with that, you're getting the water retention capacity of the peat moss and the aeration capacity of the sand. So mixing those together will give you a good mix. Now there's three important environmental characteristics that you need if you're going to be successful rooting these cuttings. Most plants will require a highly controlled environment. You need to have the proper humidity, the right amount of light, and the proper temperature. Now we're going to go over each of these separately here so you can see what I'm talking about. With humidity, it is critical. You cannot allow your cuttings to dry out at all. If your cuttings dry out, then you're not going to get roots and stem development. It's just not going to happen. Now, as I say these are with the majority of plants. There are some plants that you can just stick them into a pot, into the ground, walk away and forget about it, and you're going to get a new plant from it. And so, using this method, you will get roots on almost all plants. Even those that you can root just by sticking them in the ground, they will also root using this method. So, you want 100% humidity, 98 to 100%. You may have to intermittently mist or moisten the leaves of the plant, because they can still absorb water. But if you got them in a nice environment where the humidity stays high, once you've got it in there, you don't have to do anything until the roots form. So I like to use a moist, sealed environment to do my rooting in. Light, that's very important as well, because light drives photosynthesis. Now remember those little leaf portions that you left on the cuttings, or if you're using hardwood cuttings, they will start to develop leaves, and those leaves need to photosynthesize to start providing food for the developing roots and the developing leaves and stem on your new rooted cutting. So you want light, but you also don't want too much light, because too much light is going to burn and dry out your plants. So it's best to use filtered light of some kind. Have them in a lightly shady area, or in an area that gets maybe the first hour or two of morning sun and then goes into shade. They will do best in filtered light, but you don't want to do them in the dark. Now they will probably root in the dark, but once those roots develop and the plant starts to develop, it's going to become chlorotic, etiolated. It's going to make a weak plant. And you don't want a weak plant. You want a strong plant. So provide light to your cuttings as they root in filtered light or light shade. And then temperature. Temperature is also important. Most plants will root best around 70, 75 degrees. If you're doing them in a cool area or in a cooler part of the year, you can provide this heat by doing a heat mat or some other type of below ground or below surface heating. And so you want to provide an area where the plant is getting 70 degrees constantly, almost all day long. It's at 70 degrees, right around that range. They can root even at lower temperatures, say 55 degrees. Some plants are very adaptable and can root lower. Most plants, though, if it drops below 55 degrees, it gets too cool and they're not going to develop the roots. And plants can root the cuttings at higher than 75 degrees. Many of them will, but what you're also doing if you raise the temperature above 75 degrees, that's also the temperature that a lot of pathogens like. Even though you tried to start with sterile material, when you bring the cutting in, there's probably going to be some bacteria or fungi on it. If you get above 75 degrees in that very, very moist environment, those pathogens love it. And they're going to grow and probably devour your cuttings before they have a chance to develop roots and stems. So right around 70 degrees is the best temperature to use. And rooting chambers, as I mentioned, there are multiple types that you can do. We're going to give you a little bit of video as an additional video that you can watch later on, and how to make your own rooting chamber like this. But you can do it simple things. Like here is just a styrofoam cup with another cup over the top. Stick your cutting into the rooting media there and leave it. You've got an enclosed, high humidity, moist environment. You put holes in the bottom of the styrofoam cup so it drains easily. You pop it in there and leave it until the roots and stem develop. This is one that roots really easily. Like I said, some things you can just stick them into the rooting media, keep it watered, walk away and leave it. Just check to make sure it's moist every day. Here's another really simple one. Recycling material. This, of course, is a plastic water bottle, or you can use a soda bottle. Stick your cutting into the pot with the rooting media in it. Cover it with your bottle. Walk away and leave it. You can also cover it with a plastic bag. And then there are some plants that root very, very easily. Anyone who's ever bought fresh herbs in the grocery store, especially fresh basil, you take those home, you put them in a jar on the windowsill with water in it because you want them to stay moist and fresh. Keep that turgidity. You know that's going to make you some nice fresh herbs. Every time you want it, you just pick the leaves off. Well, if you do that, within a week or two, you're going to see roots developing down here. And you've actually taken cuttings and made rooted cuttings just by keeping them in a glass of water. Now, this is not the common way that cuttings are made. Like I said, the roots need oxygen. So with this, changing the water on a daily basis keeps your herbs fresh, but it's also allowing oxygen that's dissolved in the water to be there available to the roots as they develop. Like I said, you've probably everyone done this. If you want to root basil, this may even be the best way to do it because it happens really fast. And so this can also be considered a rooting chamber. And so now you've gotten all the information about doing vegetative propagation. Remember, label everything as you go. These examples here don't have the labels in them. They should have a label in them. Remember to label everything so you know what you got, you know when you started it, and you know then about when you should expect roots. Also, temperature, moisture, light. How important those are. So find an area where you're getting about the correct amount of light. The temperature stays about the same. Make that your rooting area. And then develop your own rooting chamber. It can be anything you want it to be. Now with rooting, I do have to go ahead and point out there are some potential problems that may develop in your rooting system. If this happens, it doesn't mean you did anything wrong. It just means that happened. Because you're growing them in an area with constant moisture, high humidity, diseases can develop. If you happen to transfer in an insect, especially a very tiny caterpillar or even an egg that's going to hatch, you put that in there, you set up your rooting chamber with that little insect that you didn't even see in there, close it up, and then you come back a couple of weeks later and none of your cuttings have leaves on them anymore. And suddenly there's a big fat caterpillar in there. That can happen because now it's in there, it's got a nice moist environment, it's got plenty of food, it's in an environment where it's not too hot, it's gotten some light, because most of them do like light every now and then, and they have plenty of food, and it's protected from predators. So that's something else that can happen. So check your cuttings carefully before you stick them into your rooting chamber. Look as closely as you can, remove any portion of the leaf that looks diseased, never use diseased cuttings, and look for little tiny insects that may be hitchhiking their way in. Algae can become a problem if it's too humid and with the light, that can become a problem. Starting with sterile media is the best way to avoid that. Start with weed-free as much as possible. If you open it up and leave it open too long, weed seeds can actually get in just from the air. So try to work with your container as quickly as possible. And remember, the light and the amount of light is very important because enclosed chambers, enclosed environments can get really hot really fast. If you got it out in the sun and it's 80 degrees outside, it's going to be over 100 degrees inside that rooting chamber. And that's going to cook your cuttings and you're never going to get anything from them. So just keep these potential problems in mind as you decide what you're going to use for your rooting chamber and where you're going to locate it. So go ahead now and try your hand at doing some rooting. That's the lab for this module. And post your results. We'd like to see how good the plants look once you do your rooted cuttings.